Hello, biology students. Today we're going to go over um, lesson one, some aspects of it. So you're going to be, first of all, you're going to be doing a mind map over lesson one, which is exploration 1, 2, and 3. You've already started doing stuff for exploration 2, but you need to read over this in more detail. Now, those of you who took physical science last year, or those students who transferred in who've taken chemistry, you've already learned about this stuff, so this should essentially be a review for you. Now, the secondary one students, since you have not had physical science yet or chemistry, you need to read over these sections very, very carefully. Nonetheless, for both groups, it would be very beneficial to have you read over these sections. So for all of us, there's a couple things I just want to reiterate, biologically speaking. So chemical reactions are taking place when chemical bonds are breaking and new bonds are forming, rearranging in the process. So this is one of the things that we modeled a lot last year. And if we were in class, we'd actually do a review activity here to go along with this. But just for right now, here's a real good example of a decomposition chemical reaction when you have hydrogen peroxide breaking apart to form water and oxygen, right? Now, hydrogen peroxide is one of these special compounds, that is really sensitive to light. Light can give it enough energy to go ahead and break it apart, and when it breaks apart, it forms water and oxygen gas. If you've ever poured hydrogen peroxide on an open wound before, you've seen this process, right? Lots of bubbles, and those bubbles is due to the oxygen gas forming, okay? So now, as a review, remember this 2 in front of the chemical equation means this is the coefficient. That means there's 2 molecules of hydrogen peroxide. The lowercase 2 are called subscripts, right, and those indicate the number of the letter or the element right before it. Okay, so that's a review. Let me scroll down. Bada bing, bada boom. So the whole point is you need to have chemical equilibrium. Right, some chemical reactions go from reactants to products until the reactants are consumed. It's like a one-way street. Sometimes the chemical reactions is reversible, and so these types of chemical reactions have an arrow pointing towards the products, right, to show you the indication of which way the equation is happening. Remember, this is called the yield sign. Okay, so moving right along. Some chemical reactions are irreversible, right? Naturally speaking, like frying of an egg, right? You can't unfry an egg in normal situations, so that's a good example of that. Some processes contain a very important aspect of the equilibrium because things change in the blood, right? So, for instance, carbonic acid dissolves in the blood. So that carbon dioxide can be transported to the lungs. And so this process is constantly changing back and forth between 2 different chemical reactions. Now, the amount of energy needed to get a chemical reaction to get started is called the activation energy. So for most chemical reactions, they need a little help getting started. Okay, so the activation energy can be thought of as a graphical analysis here, right? So you have the reactants and the products. Oh, that's the thing I forgot to mention a few minutes ago. Reactants are on the left-hand side of the equation and products are on the right-hand side. Okay, so sometimes you need to add energy in order for a chemical reaction to start, like frying an egg, for instance. You got to heat it up. Once it reaches a certain point, then a chemical reaction can then take place and produce something else. Okay, so the activation energy is the energy put in. The difference in energy is the energy that comes into the reactants to cause them to change into something else. Okay, so there's 2 types of reactions, endothermic and exothermic. The prefix endo means it's going inside. Exo means it's going outside. So in some situations, you have to put energy into it in order to get it started, like heat it up, for instance. In some chemical reactions, it takes energy from the surrounding environment to go into the reaction. Okay, so Here are the two different graphs here. So this is an endothermic reaction. You got to put energy into it to get it started. Okay, and here's an example.
Endothermic reaction.
Oh goodness. Here's an endothermic reaction where energy from the outside of the environment has to go into it before it reaches the ability to start. Okay, so pay attention to those. Now here's the part that I want to emphasize here, because this should be brand new to everybody. The concept about catalyst. And so in biology, a catalyst is a substance that increases the rate of a reaction. So it's some type of substance that helps basically speed up chemical reactions. Okay, so catalysts neither change nor consume during a reaction. This is an important concept to know. So they're technically not part of the equation, right? So we don't account them as part of the equation. All right. So catalysts provide an alternate way for the reaction to occur that requires less activation energy. So that's kind of the whole point. Once you introduce a catalyst to a chemical reaction, it decreases the amount of energy you need to put into it before it can get started. All right. So this is kind of important. So in biological terms, we have catalysts that are called enzymes. Now, enzymes are catalysts that lower the activation energy and increase the rate of chemical reactions. This is both reversible and irreversible reactions. So enzymes are involved in almost every process, from breaking down food to building proteins. Okay, for example, digestion, an enzyme called amylase is in your saliva that breaks down starches in food. Okay, so that's the enzyme that's in your saliva. In the intestines, other enzymes called maltase break down sugars like maltose into glucose molecules. All right, like once again, if we were here at school, we would have a little lab activity that we could play around with this, but unfortunately, we don't have that ability to do that right now. So a couple other things that need to be important. This is a real big concept. The enzyme structure is important because it allow only certain reactants to bind to the enzyme. All right. This is called the activation site. The specific reactants in that enzyme act as substrates. Okay, this is the most important word here in this section. Be sure you understand what this word means. The substrate is the thing that the enzyme is bonding to. Okay, it is the thing that the action is being done to. Okay, it's like the enzyme is the verb, so to speak, and the substrate is the thing that is happening. Okay, the action is taking place on it. Okay, now the substrates and enzymes, they work together in a lock and key format. Okay, just like a key fits a certain type of lock, the substrate fits to the activation sites of the enzyme. All right, this is why if the enzyme structure changes, it might not work well at all. Now, enzymes in biology are very important because they're heat sensitive. Right, if it gets too hot, they can change the shape. The enzyme doesn't work anymore. If it gets too cold, it can change the shape, and the enzyme doesn't work anymore. All right, so here's a good example of the lock and key model. So the enzyme here is maltase, right? And the substrate is called maltose. Maltose is the thing that's being broken down. As you can see, maltose is basically two glucose molecules bonded together. All right, and the enzyme here is in the shape of the substrate. All right, so the sugar maltose is broken down once it comes in contact with this enzyme. Right, it's a lock and key. Right, you can think of this like the lock, and you can think of this part like the key. They fit together. Okay, now once they fit together,
the maltase enzyme is shaped so that only the maltose molecule fits into the active site of the enzyme.
Yeah, so this is really important. So if it was 3 glucoses here, it wouldn't fit with this enzyme. It only fits with 2. All right, now once it fits together, the activation energy is there. Now it can be broken down further, right?
The enzyme allows a chemical reaction to occur that breaks the maltose molecule into two glucose molecules.
All right. So basically, all enzymes operate this way, and there's a specific substrate that fits with a particular enzyme. Okay, and so almost all enzymes are proteins, all right, on the exception of a few. So what you're going to do here is you're going to make your next assignment is you're going to make a three-dimensional model of an enzyme and a substrate. Okay, and I'm going to post this video on this assignment for you. Okay, so you can read over the last sort of part here, but the key important functions you have to understand. Enzymes are pH specific, and they're also temperature specific, right? If the pH changes or the temperature changes, it can affect the shape and the function of the protein. Okay, this is why if you run a really high fever, it can be deadly because you have lots of enzymes in your cells, right? If your temperature gets too high, it can cause them not to work properly. And so, and that's the case, and your cells can't function properly, and that can lead to an imbalance of homeostasis. And next thing you know, boom, you're dead. So that's not good. All right. So you can look over the rest of that. But nonetheless, these are the two assignments you're going to work on this week is the mind map and also the three-dimensional model of these. Okay. 3D means it's three-dimensional, not drawn on a piece of paper. You got to make something. Good luck. Let me know if you have any questions.